possibility, at least I didn't know when I was setting up my dissertation work. This is what I wanted to test, because this gives qualitatively different dynamics, because if we start most with the broadleaf grass, now it changes the composition of the mutualist community in a manner that the narrowleaf grass now has greater fitness. So the narrowleaf grass increases in abundance till it reaches dominance, and then it reverses back. So this is a dynamic that can lead to coexistence. It also minimizes the benefit that any one plant receives from its community mutualist over time. So it sort of illustrates a potential conflict in the long-term stability of mutualisms. So I wanted to test this. I did it in this field, and that ended up using whole soil, and I sort of failed because I couldn't convince myself that I was looking at the effects of mycorrhizal fungi. Then I got a USDA postdoc, and I learned how to do mycorrhizal fungal work, and I then isolated the mycorrhizal fungi from that field. Here's 9 spores that were combined. They were single spore. They were clean cultures that I then mixed together to make a community picture here. All of this is the diameter of this is big enough to see by eye, so is these ones not so much. So these are large, single-celled, multinucleate. Individual cells are genetically diverse, and that's going to be an issue that comes up later in the talk. But with this clean cultures that's now free of mycorrhizal fungi, I went to the effort of starting them from single spores. I then did the feedback test, started with mixtures of the mycorrhizal fungal communities, allowed it to differentiate for a growing season. I tested, I monitored the differentiation of the community. I put them back in the same pots, trained it again, so 2 growing seasons, and tested and monitored and tested again. And what came out of that is, so I found overall negative feedback between this species and this one, and this species and this one, and I ended up just focusing on this one to dissect all of the individual effects, and this is what it looked like. So panicum does best with this species. This fungus grows best with Plantago lanceolata. Plantago lanceolata grows much better with these 2 species, and these 2 species grow better with panicum. So that generates a negative feedback dynamic, which is interesting because it could be a mechanism for the maintenance of diversity of the fungi and the plant. And it's interesting because it could be a mechanism for facilitation. This is a native plant. This is not native. So this native plant is facilitating the fitness of the non-native by increasing the fungi that it does best with. And it's interesting because it illustrates that over time, the benefit that plants are receiving from their mycorrhizal fungal community is declining. And so that identifies the challenge of understanding what are the stabilizing forces in mutualism. And I'm not going to tell you, I've done work on that, and I think it's super exciting, and I'm just tell you that that's the 3rd talk. So it's, okay. Now I'm going to go back to what we talked about yesterday. So soil pathogens as drivers of coexistence in this system. But then I talked about how if you take the seeds from this and you put it in post-agricultural soils, you get a dominance of one species, low diversity. So maybe that's due to loss of pathogens. I think that could be a big part of that. But when I showed you that we could move the seeds with the microbiome and then increase the establishment of this diversity in this post-agricultural soil, I was illustrating it with my picture of mycorrhizal fungi, and that wasn't dishonest. So I'm going to talk about the evidence that this native mycorrhizal fungi plays an important role in the persistence of diversity in this system. But it's not due to the stabilization forces. It's not due to the coexistence process. It's due to the process of making species possible, making their growth possible in this environment. So making it, increasing the pool of available species. So to make that point, so first, let me introduce mycorrhizal fungi. So, okay. This is the structure inside the cortical cell of the roots. It's sort of an invagination of the plant cell membrane, and then the fungal cell membrane is pushed up against it in this highly branched structure to increase the surface area between the fungus and the plant. And then this is called, they're called arbuscular mycorrhizal fungi because of that structure. That's the structure that involves exchange of resources from the fungus to the plant, exchange of resources from the plant to the fungus. So most plants, including corn, associate with this fungus, and this fungus has hyphae that goes through the soil, and it's good at acquiring resources from the soil, particularly phosphorus, but also nitrogen and water, and delivering it to the plant. The plant delivers energy in the form of lipids to the fungus. It's been shown to modify plant defense. These are important fungi in stabilizing the soil and preventing erosion. They're all-around good guys. But as I showed you, they can be ecologically diverse, and these different fungi have different functional properties. And we know that because we grow them, because we isolate them and grow them. And so these are different fungal species. These are they're identified by their wall structures here that you're seeing here in electron micrographs and light microscopy. And from growing them and testing them, we know now that, let's see, maybe I'll just go with this, that they differ in their overall effectiveness on plant growth promotion. They can differ in the host specificity of plant growth promotion. One fungus might promote one plant more, another fungus promote a different plant species more. They differ in their effect on host defense, in their ability to hold soil together, and because of that, the composition of mycorrhizal fungi will be an important thing that we should care about for a terrestrial ecosystem function. But they also differ in their dispersal ability, in their competitive ability on plant roots, in the host specificity of their growth rates. One fungus will grow better on one plant than the other, in their seasonality, in their sensitivity to disturbance, in their sensitivity to soil factors. And because of that, the composition of mycorrhizal fungi is going to change over time. It's dynamic. And the challenge to understand the long-term benefits or the long-term influence of mycorrhizal fungi in community function is to understand how this changes this, right? This intersection of these two, of the forces driving the dynamic of the fungus with the forces driving the benefits of the fungus. And so if we just focus on the host specificity effects, that's the focus, that's the feedback story, and I'll return to that feedback story at the end of the talk. But the restoration story is due to all of this. All of these things, including disturbance, changes the composition of the mycorrhizal community, and then it has cascading effects on the functioning of the system. And so I'm going to talk about that restoration story in the context of the tallgrass prairie, which is highly diverse, which is then converted to agriculture. And then if you abandon agriculture, you get a diversity of mostly non-native, but also native weeds. And then if you take the seeds from this and you put it in this environment, you get this low diversity tallgrass. And this is one of the older restorations at the Fermi National Accelerator Lab near Chicago. So standard restoration practice failed to restore the original composition, and that's the restoration challenge in the context of this system. We easily get dominance of the natives. That's sort of different than a lot of Western systems where you struggle to get the natives to dominate. But the missing species are not random. So if you arrange species on a life history gradient, so you have fast growing, rapid growth, high allocation to seed, you got these annuals, biennials, fast growing species, then you have very slow growing, long lived, late successional species. These are the species that are missing from the restoration. Not all of them. The tall grasses are late successional species, but the vast diversity, there's a great diversity of these slow growing species in the tallgrass prairie system. Those are difficult to reestablish in restoration. So, okay. So in this talk, we're going to focus first on the differential response of prairie plants to mycorrhizal fungal species as a way of answering this first question. do AM fungal mediate restoration success? And then we'll look at the degradation of the mycorrhizal fungal community, and then we'll look at reintroduction and does that the tests of the restoration benefits. So, and then we're going to ask, what is the overall fungal dynamics and how does it mediate plant succession? So they're overlapping goals, but this plant succession depends on the natural dynamic and particularly the differential influence of prairie plants on mycorrhizal fungi, because it depends on the feedback dynamic. And so we'll talk about that, and then we'll talk about the implications of this feedback dynamic for the restoration goal. Okay, so late successional species are missing from restoration. So the first thing we're going to do is ask if these late successional species differ in their interaction with mycorrhizal fungi from these early successional species. So we have an array of species, annuals, slow-growing, long-lived plants, and we're going to test them with and without mycorrhizal fungi in the greenhouse. And so that's what this is, this mycorrhizal response. It's the ratio of growth with and without the mycorrhizal fungi. So when that ratio is 1, they are not growing differently. That's growing the same with or without mycorrhizal fungi. Each dot is a different species of plant. And so we have early successional species, very early, middle, late successional species, the way we divided it up this way. And these late successional species are on average 6.5, 7 times bigger with mycorrhizal fungi than without. And there are some points off chart up here 20 times bigger with the fungus than without. So these late successional species grow better with mycorrhizal fungi. They care more than these early successional species. And so we can then look, this is a test of like, I don't know, 30 some species. We've now collected data on around 200 prairie plant species and prairie of this area, including non-natives, early successional native, mid-successional, late successional native, and it's a very robust pattern. So consistently in the Midwest, non-natives and early successional natives similarly have low responsiveness to mycorrhizal fungi, while late successional and mid-successional native plant species are highly responsive. So that's just overall response. If we look at how variable that is and how it depends on mycorrhizal fungal species identity, that's what we're illustrating here. So we have 6 early successional species here, and in this study, they ended up doing worse with mycorrhizal fungi than without, and 6 mid and late successional species here, and this is on log scale. So then we have plants that are, we had, I think, what is that? 7 different mycorrhizal fungal species. And I want you to look at the variation. So they're arranged in terms of the most responsive on average to least responsive. So the early successional species, not a lot of range, and it doesn't matter much what the fungus is. These mid and late successional species, high variation in how responsive they are, depending on the identity of the mycorrhizal fungus they were inoculated with. So we can represent that statistically as the coefficient of variation, the ratio of the variance over the mean. And the late successional species have greater sensitivity to the identity of mycorrhizal fungi than the early successional species. And that's a test of 12 species. If we look across, if we do more studies and look across all the data available, the non-native and early successional species have low sensitivity to AM fungal identity, while the late successional species have much higher sensitivity to AM fungal identity. So late successional plants are missing from restorations. They're more responsive to mycorrhizal fungi, and they're more sensitive to AM fungal identity. So are AM fungal communities degraded by 100 years of agricultural tillage? And so to answer that, we went to prairies, remnants, and nearby post-agricultural grasslands, and we sampled the soil, and we sequenced out, we amplified out mycorrhizal fungi, and looked at the change in composition with disturbance and across the precipitation gradient. And what we found was that there are consistent differences between prairie remnant fungi. So these dark ones are significantly more abundant in remnant, and the dark ones over here, pointing this way, are significantly more abundant in the post-agricultural grasslands. So there's a shift in composition that's common across this entire range, where we have prairie fungi, and then we have weedy post-agricultural fungi. So this is evidence that AM fungal community is degraded with disturbance, and it's consistent across this entire range. But we looked also whether they have specialization to wet versus dry, specialization with climate, and we found it for remnants. So remnants are differentiated in remnants. If you just look at the undisturbed remnants, then you have one group of fungi that's common in the higher precipitation part of the prairie, and a whole separate group of fungi that are common in the drier part of the prairie. If you look in the post-agricultural sites, you don't see that. They all are looking basically the same. So this pattern here is consistent with local adaptation to climate. We didn't show it, but it looks like that if that pattern is local adaptation, we lost it with agricultural disturbance. Okay, so we have different compositions following agriculture. Then can reintroduction of the native mycorrhizal fungal community accelerate succession and improve plant diversity and the composition? So then we have the challenge of how do we reintroduce the mycorrhizal fungi into this site? And we started doing this more than 20 years ago, and I took buckets of dirt from an area that was being lost, a prairie that was boxed in by factories and was going to be lost because it couldn't be burned anymore. And I dumped them into post-agricultural soil and I tilled it in. That worked. And then we also can inoculate nurse plants and demonstrate that and sort of look for them to spread to neighbors, and we see that it does. But in both of this case, if we're inoculating prairie soils, then we're raiding the undisturbed sites. And so the more sustainable approach is to isolate and culture the native mycorrhizal fungi. Then we can try different approaches to reintroducing them into this. So we've done, I don't know, 20 different inoculation studies. I'm going to show results from two different studies. The first one demonstrates that mixed isolates of native AM fungi perform as well as whole prairie soil. And the next one looks at the benefit as a function of inoculation density of these prairie mycorrhizal fungi. So this first study, it was funded by the Department of Defense. They do training in grasslands, and they need to restore the grasslands after they drive tanks over it so they can drive tanks over it again. So they supported us to evaluate the benefit of mycorrhizal fungi in grassland restoration on Department of Defense properties. And so we had 5 restorations at these 3 different locations, and we consistently, across all of them, we inoculated with native nearby prairie soil, we cultured out nearby native mycorrhizal fungi, or we didn't inoculate. Those are the 3 treatments. And each of these sites had a very different land use history and site preparation. Some of them used solarization, some of them used herbicide some of them didn't have site preparation. And at each site, we tested 4 mid or late sessional prairie species, and there were different species at every site. And we inoculated them with these different treatments, and they were planted out as plugs, and we also looked for spatial spread from those plugs. That was the overall goal of this. And so I'm going to show you the overall result that was common across all 5 restorations is that survival was greater when they were inoculated with native mycorrhizal fungi or prairie soil than the control. That was survival in the first year. Survival in next year was also greater, and they grew bigger if they were inoculated with the native prairie soil or native mycorrhizal fungi. So what that looks like, if you put out the same initial composition of plugs, then at the differential survival in the first year looks like this, the differential survival in the second year looks like this, then the differential growth rate looks like that. So the whole thing gets you closer to looking like a remnant compared to the standard restoration practice. So the next test, we just have mixtures of native mycorrhizal fungi. This is in Kansas, and we have inoculation densities from 0 to very high densities equivalent to those buckets that I dumped in that first test. And so we have 8 different inoculation densities, and then these are split plots. This is the experiment here. We have split plots where half of them are rows of the Land Institute candidate perennial crops. This was funded by the Perennial Agriculture Project. And the other half is just a diverse prairie seed mix. And we're mostly going to talk about this side of the plots, but it's worth noting that these candidate crops were grown, inoculated in the greenhouse, and then they were just planted out here into these plots that have different densities of mycorrhizal fungi, of native mycorrhizal fungi. And the response to inoculation was predicted. The response that we got in the greenhouse for those candidate crops predicted how responsive they are to native prairie fungi density in the field. So confirming that the work that we're doing in the greenhouse is meaningful for what we see in the field. So this is the result in the field. This is the native richness in that first summer following the initiation of the project of the plots. So we have greater native plant richness and greater native plant abundance in the first year. After 3 years, the native diversity is still high, the native with greater native abundance of grasses and legumes, and we have we're having reduction in the diversity of weeds to having basically no weeds in the high density with high native prairie density. And then after 7 years, so here we're getting to what is valued in prairie restoration, which is the high plants of high conservation value. So we're looking at what is the abundance of high value plants? And the abundance of high value plants, these are late successional plants, increases with the density of inoculation. And the floristic quality index, which is a function of the density of these high value plants and overall diversity, also increases, and the total diversity increases. In the difference here at the top, this is the plots with seeded, and then here we're showing the diversity in the unseeded plots. This was originally row agriculture with the same density. And then this prairie seed coming in is just not initiated. It was just coming in from neighboring locations, but they are still getting benefits. So even if you don't put the prairie plants on, there's, much weaker benefits of inoculation, but they're there. So that's the restoration story. Late sectional plants missing from restoration. Those plants that are missing are more responsive to mycorrhizal fungi, they're more sensitive to AM fungal identity. AM fungal communities are degraded by disturbance. Reintroduction of these native late sectional mycorrhizal fungal communities increases the establishment, growth, and suppresses weedy plant species in restoration. Okay. So all of this is built on just knowing the response of late sessional plants compared to early sessional plants to mycorrhizal fungi. So this part of this relationship. But the successional dynamic in the influence of mycorrhizal fungi on the plant community is going to also depend on this part of this relationship. How do these fungi respond to late successional plants compared to early successional plants? So one way to think about this, and this is the part of the stability of mutualism lecture that I'm not giving, but I'm giving you this result, which is what we do is we take one plant and we split its roots into two different independent pots. One pot is inoculated with beneficial fungi, and one pot is inoculated with non-beneficial fungi. And we label the carbon in this plant, and we look at how much carbon goes to the good fungus versus the bad fungus. So if the plant can preferentially allocate to the good fungus, that's the way it can keep its fungus beneficial, it can stabilize the mutualism even when delivering benefit to its host is costly. So this is done in part of a larger test of processes that maintain the stability of mutualism. And what I'm going to show you is the ability of the plants to preferentially allocate to the most beneficial fungus as a function of plant life history, from these earlier successional native prairie plants to the later successional native prairie plants. And so here, this is Rudbeckia hirta, Tradescantia ohioensis, Elamis canadensis, early successional prairie plants, Echinacea pallida, Liatris, spicata, Allium sinuum, and little blue stem here. So consistently, the early successional plants are not able to allocate preferentially to the best fungus, while the late successional plants are able to allocate preferentially to the best fungus. So from this, we would expect that these late successional plants should be increasing the best fungus in their roots, and that should generate a positive feedback. And so to test that, I showed you how I tested that overall feedback through mycorrhizal fungi in my postdoc. And we just did it, but we did it at a larger scale. So we have lots of plants ranging in life history and phylogeny, and we grew them for a period of time. We actually grew them for 2 growing seasons again, and then we do the full factorial test for feedback. And they're initiated, well, we started with 38 different plants, ranging, so phylogenetically diverse groups, and a range of life histories from early and late successional life histories. And we started them with these 7 mycorrhizal fungi, which we've characterized. These are the same 7 that I showed you that first figure. So we know a lot about how the plants respond to these 7 mycorrhizal. They're all from prairies. They're all from prairies from the Illinois, Indiana area. And this is the first result. So here we're looking at whether so each of this is the abundance of each different species. Is it off chart here? Oh yeah, here it is. So these are the 7 different species are over here. The total diversity is here, and the proxies of the density of mycorrhizal fungi are here. And then we have tests by plant life history and tests by plant phylogenetic structure. So we have significant differences in composition, and we have significant differences in AM fungal diversity that's structured by plant phylogeny and by plant life history when we take out that phylogenetic effect. It's a bit of a mess to look at, but those are the take-home stories from that. But each of these fungi is genetically polymorphic. They all have every isolate has many, many nuclei, and those nuclei are genetically different. In fact, they're different at the ribosomal gene that we're using to monitor their abundance. And so what I showed you before is grouping all of the amplicons by species and looking at the ecology. This is looking at the genetics. Here's the different fungi, different fungal species are different colors here. And then we're looking at whether there are different genetic compositions as a function of phylogeny and plant life history. And it's not unusual that the genetic composition is changing with hosts over the 2 years that we grew them, and that genetic change is structured by either phylogeny or life history, which is going to make the interpretation of any feedback difficult, because is it due to ecology or is it due to evolution? But so to test feedback, we didn't test all of the combinations. We did a sort of a subset of the possible combinations. And then we looked for patterns of the subset that we did test. So we're always looking at, for every pair, we're looking at how they do with their own mycorrhizal fungal community compared to how they do with each other's. And so overall, the overall feedback ranges from significantly negative to significantly positive for individual pairs of prairie plant species. So the phylogenetic distance between those pairs of plants doesn't really predict. This nonlinear relationship is not significant to 0.05 levels. It is less than 0.1, so I will leave it up to you on whether you say phylogeny does or doesn't predict that relationship, but I don't have a priori reason why this phylogenetic distance would produce positive feedbacks relative to these. So I'm inclined to just say there's no relationship. But when we look at differences in life history, we do see consistent patterns of feedback. And particularly, we're interested in this one. When we pair early and late sessional species, this is what we were started off caring about, and we get consistent positive feedback. This outlier actually is in that life history category, but even with this point, the average is significantly positive. The median is significantly positive. No matter which way you do it, it's positive. We're also getting positive feedback amongst when the two plants are in this early successional category. And that says that this early successional category actually represents very different life history patterns that's not simple. It should perhaps be dissected out more, that some of them are good hosts and responsive and some of them are not. We don't see significant feedback when both of the species are late successional, and that's what we expect. Late successional species, if they're all good hosts and they're all responsive, then they don't care whether it's their neighbor's mycorrhizal fungi or their mycorrhizal fungi. They're happy, and that's what we see. So all of this is consistent with the dynamics of the fungus contributing to turnover during succession. So yeah, we'll return to that. So going back to this, is it ecology or is it evolution? So we can look at the dissimilarity in species composition between the two plants that we measured the feedback on, and does that dissimilarity in species composition predict do pairs with greater dissimilarity generate greater positive feedback? That's what we'd expect. Specialization and symmetry and fitness relations should generate stronger positive feedback. We don't see it. So we don't see any evidence that the differences in species composition is generating the feedback that we saw. But when we look at the genetic composition, two of the common species, Entrophosphorum frequens and Chlorioglomys lamellosum, the differences in genetic composition, when you're more different, you get greater positive benefits for both of these, in both of these scenarios, for both of these fungi. So that says that these fungi, even though they're in a mixture, and we picked these 7 fungi because they affected the growth of early and late sessional plants differently, the change in composition, which we set up to be meaningful, was not meaningful. Instead, the evolution within two of these ended up being the major driver of the functional change that we saw, which is not what I expected, and sort of not a little bit of a challenge for me because I grow mycorrhizal fungi and I have to grow them on live hosts, and within 2 growing seasons, they're functionally changed depending on what host I'm growing them on. So what we see is that late sessional species are more responsive to AM fungi and they're more sensitive to AM fungal identity, and late sessional plants are better hosts for beneficial mycorrhizal fungi. So we get a consistent positive feedback between early and late sessional species that can accelerate succession, or it could inhibit succession. And so to be clear, positive feedbacks can generate alternative stable states. So one of those stable states is late successional dominance. The other stable state is weed dominance. Late successional dominance with good fungi, weed dominance with bad fungi. Those are alternative stable states. This is the way it's commonly illustrated, where we have a desirable state, highly diverse late successional plants. We have a degraded state, non-native and native weeds, bad fungi. And in this region here, we have repelling forces which make this stable or this stable depending on where we are. And it'll depend. So that is the way to operate this on this system. So positive feedback can inhibit succession when the beneficial mycorrhizal fungi are rare or absent. So if we don't have beneficial mycorrhizal fungi in the system, we could get stuck down here. But if we do have beneficial fungi, then the same dynamic can accelerate movement up to this state. And this expectation is the mean field expectation. So the mean field means like thinking about the dynamics if everything was in a aquarium, in well-mixed aquarium. Plants aren't like that. Plants are spatially structured. They're interacting mostly with their neighbors. And when you consider that, then the actual dynamics are going to be a little different here. So here we're blowing this up and we're thinking about 3 points. If you're out here, it doesn't matter how big your patch is. You're going to be stuck there. Small patch, big patch, you're going to this state. When you're down here, it doesn't matter how big your patch is. Small patch, big patch, you're going to go back to this state. So that's what that stability says there. But here, where you have the alternative stable states, in this condition, the dynamics in where you go will depend on the size of the patch. So if you have a big patch here, then you have the possibility of that patch growing, and that would get you up to the desirable state. But if you put in a small patch, there's a likelihood that all of the dynamics are determined by the local scale abundance at these edges, and you could be driven extinct. So this process is called nucleation. It's a common process that people in chemistry, they think about it in physics, about the process of crystallization. It's a spatial process because what matters in the growth is the relative abundance at the edge of these patches. In the interior of the patch, it doesn't matter. It's all blue. At the outside here, it's all green. And it's all local dynamics at the edge, and that determines this size of the patch mattering at this point. And so the fact that we have positive feedback driving the quality of restoration and driving success of succession then indicates that one strategy for restoration is to manipulate patch size and try to get the patch, bring in patches in the environment that actually will spread, that will nucleate and increase over time. And that is something that we're testing in this system, and we have evidence that it does work, but actually seeing the patch grow like this is going to take a while. Okay, that's all the science that I have. And I wanted to end with like, just what does it mean? So, what we showed is that having native mycorrhizal fungal isolates can be useful to conservation biology because a lot of these late sexual species are rare or endangered, and restoration biology. And so we also know that mycorrhizal fungi are locally adapted to their soil and climate. I just described the study where we showed differences with climate. People, when do reciprocal transplant combinations, they can show that they do better with their local conditions. And that's what we know from the plant side. Having local plants people put a lot of work into getting local seeds for their restoration. This suggests that we should put an equal amount of work into getting local mycorrhizal fungi into your restoration. And we have a lot of isolates. We have 35 clean isolates from eastern and western prairie remnants that in our lab. This is our lab collection. It's a lot of work maintaining those. And what I want to talk briefly about is the challenge of, so to succeed in this, to successfully apply this, would require a big investment in collecting and isolating mycorrhizal fungi and maintaining them. And we maintained these ones, and we then went about adopting another huge collection. So we got super exposed to the burden of maintaining a collection. So the International Culture Collection of arbuscular mycorrhizal fungi was started in 1985 by Norm Schenk at the University of Florida. He built the collection up to 182 isolates with 56 species. And then Joe Morton at West Virginia University, with Bill Wheeler helping him, built the collection up to 1,200 isolates and 103 species. That was Joe's estimate. So my wife Peggy and I learned about how to work with mycorrhizal fungi from Joe. So when Joe retired, the West Virginia University didn't hire anybody to adopt the collection. And so Peggy and I adopted the collection, which now has 63 really identified species, 916 isolates. That represents the entire span of genera that are now recognized, and representing many continents many countries and continents on the Earth. And so we've had it at KU since 2021, and Liz and Tara do a lot of the work in maintaining it. And we rescued it initially with funding from the National Science Foundation. But just to go through what we do, so every culture is grown out each year on live plants. So this is what that greenhouse operation looks like. So that means they're then air dried, stored in the cold room. And so that means that every week we're starting about 20 isolates in these 20 pots, and then we're harvesting 20 pots and checking that they're the same spores as what is supposed to be there. So this is a ton of work. We moved it with support from the NSF to save the collection. That support ended in last May. And so the long-term financial security of this collection is quite uncertain. We do distribute them. It's a service for research. We distribute to anybody who needs the mycorrhizal fungi or has use for them. We do charge fees for that, but there's no way that we could charge enough to actually maintain the collection itself. So we're sort of in this worrisome state of securing this resource for investigating mycorrhizal fungi. But I will say that with what I just showed you, that the genetic composition changes in a meaningful way depending on what host plant it's grown with. Joe used sorghum grows, and we still use sorghum because Joe used sorghum. And they've been maintained on sorghum for 40, sometimes some of them as many as 40 generations. So, and it is then questionable whether like this would be a resource for conservation biology or restoration. That's something that we can't answer. We're now testing whether whether isolates from different origins function better in that site of origin as you'd expect, and that's something that we're looking at now. For our prairie isolates, we've always maintained them on a mixture of prairie plants, so we've rotated those plants every generation. So we sort of have been avoiding this convergence on sorghum mycorrhizal genotypes by doing that. So I think that's all I got. And so I just wanted to open up a conversation, about how if this is a useful tool for restoration and for conservation, for plant conservation, then it would take an investment in creating the resource for restoration practitioners to apply it. So I'll just flag that Liz has taken our lab isolates from prairies, and she started a company so she grows them up as sort of a weekend warrior activity in large quantities. It's called MycoBloom. And so that is our lab isolates that we've shown are really good for tallgrass prairie restoration, and you can buy them and apply them. And she is involved in a big Teddy Roosevelt library restoration. So that is now being used on relatively large scale, but not as much as you might hope. So anyway, thank you all for your attention. I'm happy to. Yeah.
This history of plant soil feedback. Where do soil aquifer?
Well, I'm sorry.
Soil aquifer committee in this story. Do they not have any impact on this feedback?
I'm sorry. I have bad ears, so I'm still troubling.
So I was asking about the role of soil aquifer committees in this whole plant soil feedback story.
Just the mineral part of the soil.
The soil aquifers.
Yeah, the soil. Yeah, the
whole soil biodiversity, not only just AMF and other fungi or bacteria.
Yeah.
But the whole.
Yeah, right. It's massively diverse. Exactly. So I agree. I don't know. So I focused on pathogens and mutualists because they have strong direct effects. And so a lot of the biodiversity in the soil is driving processes like nitrogen turnover, a lot of different decomposition. Those I would expect are going to be more indirect effects and more long-term effects on plants. So when I've tested them, so when in the experiments I talked about yesterday, where we have feedbacks from these field soils, we tested differences in bacterial composition, saprotrophic fungi, difference in none of them were significant predictors of the actual feedback. So I think they're super important to soil ecology. How that ties back into actually altering differential plant response, because if they raise the growth of everybody, like increasing nitrogen, then it's not going to be a differential response, and it won't. The net feedback, the net effect on plant interactions will be zero. And so so right now I would suggest that they not that they aren't important, but they aren't as important as the direct effects of pathogens and the direct effects of mutualists.
All right, let's thank Jim. He'll be around if you have any more questions. You can come up and ask.
But you showed that towards the end, this alternative stable state.
Yeah.
That the positive feedback can keep them in a weedy state or a late successional state.
Yeah,
But you also showed that the late successional species are better responders and better hosts. So don't you expect it, isn't the late successional state the more likely outcome?
yes. So if the good fungus is available, if the good fungus is available, it should increase in that I agree. And that is the case with succession, right? So that is sort of, it's not that, but this